Running is a part of our everyday lives. We participate in or watch short and long distance running. However, the question we forget to ask ourselves is, are these races fair?
Oh, they're away, and Gatling got away brilliantly, and he's ahead of the field at the moment, and Bolt going brilliant. Here comes Usain Bolt! Usain Bolt storming through, he takes it again, Blake gets the silver, 9.64!
Welcome to the genetics of running. We all know how big of a role physics plays in sports. The forces acting on a runner are the forces of gravity and the x and y components of the normal force, also known as a ground force. Also, we can use the kinematics equation delta t equals v1 delta t plus half a delta t squared to calculate the runner's acceleration. The acceleration is 2.03 meters per second squared. However, many people don't know that biology plays a huge role in who wins a race. Why are certain athletes able to outperform others? Well, it's all in their genes. No, not those genes. These genes. A gene is a part of the chromosome that governs the expression of a specific trait in an organism. The law of segregation proposed by a famous geneticist, Gregor Mendel, concluded that there must be pairs of hereditary factors from each parent that determine the result of each gene. These factors are referred to as alleles. Therefore, alleles can take alternative forms. For example, there can be one allele for the yellow pea color and one allele for the green pea color. Both alleles code for the gene that determines pea color. Mendel also proposed the law of dominance. This law proposes that some alleles are dominant while others are recessive. In genetics, scientists represent dominant alleles using uppercase letters and recessive alleles using lowercase letters. The combination of alleles in an organism is known as its genotype. The expression of a genotype that can be observed is called a phenotype. Studies done by scientists have shown that many pro athletes have genes that allow them to have a fast metabolism, large muscle size, immense strength, strong muscle fiber composition, high anaerobic threshold, large lung capacity, high flexibility, great stamina, and high endurance phenotypes. But the question is, how did they get these amazing phenotypes? The answer to this is meiosis. Meiosis is cell division that occurs only in reproductive cells, known as germ cells. In meiosis, there is meiosis 1 and meiosis 2. The end result is 4 cells with one set of chromosomes each, called haploid cells. In mitosis, the end result is 2 diploid cells. Diploid cells have 2 sets of chromosomes in them. Before meiosis begins, the chromosomes inside the cells begin to duplicate. The first part of meiosis 1 is prophase 1. In prophase 1, DNA coils around tightly and is visible under a microscope. Homologous chromosomes, chromosomes that code for the same genes, become closely associated in synapses, and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has dissolved and the microtubules form a spindle. Spindle fibers connect to one side of each centromere, and the 2 homologous chromosomes attach to the microtubules, orienting from opposite poles. Now, each pair of chromosomes line up on the metaphase plate. Either a maternal or paternal homolog may orient towards a given pole. In anaphase 1, the microtubules of the spindle fibers shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains 2 sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the 2 sister chromatids are not identical. In cytokinesis 1, the 2 cells separate. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers attach to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move to opposite poles. In telophase 2, the nuclear envelopes reappear around the sets of daughter chromosomes. In cytokinesis 2, the four cells separate. That is the end of meiosis. After meiosis, the sperm will fertilize the egg. In fertilization, the first sperm to attach itself to the outer membrane of the ovum will release its genetic material into the ovum. The ovum's pronucleus and the sperm's pronucleus attach, giving the zygote 46 chromosomes, 23 from each parent. At this instance, all the genotypes for the zygote are determined and the genome is formed. Sometimes genetic mutations occur and an individual will have an incorrect number of chromosomes, referred to as a monosomy and trisomy. This causes disorders such as Down syndrome and Turner syndrome. However, not all genetic mutations are detrimental for the organism. In reality, mutations are beneficial because they allow for greater genetic diversity. Genetic diversity is key because it increases the ecosystem's ability to adapt to various conditions. A study by the Journal of Strength and Conditioning Research published by the National Strength and Conditioning Association shows that mutated genes AGT and ACTN3 are 2 to 3 times more common in power athletes. Power athletes include runners, weightlifters, and jumpers. They discovered that the C allele of gene AGT is more common amongst power athletes. 55.5% of power athletes had at least one copy of this C allele. This means that many athletes participating in professional running have performance enhancing genes. Also, many scientists have noticed that certain ethnic groups perform better in professional running than others. Well, the reason for that is the theory of evolution. The theory of evolution can be explained with biologist Charles Darwin's study of finches. On Darwin's trip to the different islands in the Galapagos, he noticed that each island species of finches had different types of beaks best suited for the food available on the island. Therefore, he suggested that a process that scientists now call natural selection must be taking place. It's a process by which organisms that are better adapted to their environment tend to survive and reproduce more. Therefore, mutations that allow for better adaptability tend to prosper. Therefore, the same can be said about genetic mutations that allow for better athleticism. Many ethnic groups reside in environmental conditions that require traits such as high speed, high strength, and high endurance. Only humans with adaptable genes are able to survive. That's why certain ethnicities are faster at running than others. They receive these traits through natural selection because only the athletic humans in those ethnicities were able to adapt to the environment. The Spanish scientist that discovered the alpha-actinin 3 gene, also known as the ACTN3 gene, determined that this gene allows humans to contract muscles at extremely high speeds. This allows for an extremely fast breathing rate and an extremely fast rate of cellular respiration. The rate at which the diaphragm and the intercostal muscles contract determines how fast the human breathes. This links to how fast a person is able to convert glucose, broken down by the digestive system, into energy. The oxygen obtained from the respiratory system and the glucose obtained from the digestive system is used to create adenosine triphosphate, also known as ATP, and carbon dioxide plus water as byproducts. The body uses ATP for energy. The rate at which the body will perform cellular respiration is much faster in those with the ACTN3 gene, thus allowing for much faster muscle contractions that help in moving at high speeds. The scientists discovered that athletes without this gene have muscles that are more susceptible to fatigue. Having this gene gives runners a drastic advantage in races. However, the AGT and ACTN3 genes are not the only factors that determine a runner's athleticism. A healthy diet with the right quantity of macronutrients and a good exercise routine can help an athlete's muscles develop. Also, many athletes dope using drugs such as steroids as a way to increase performance in an unethical way. In the recent 2016 Rio de Janeiro Olympics, the media and International Association of Athletic Federations brutally criticized South African sprinter Caster Semenya by calling her a man. This was because she had high levels of testosterone despite being a female. They thought she might have taken hormone increasing drugs or may have gotten surgery to increase levels of testosterone. Therefore, there was talk about how the Federation was going to ban her from participating in races. This leaves us to one question. Do you think that athletes with genes that enhance running performance should be banned or placed in a different category than those who don't have those genes? What's your opinion? Please do the survey in the description below to help with my research. Thank you for your time and I hope you learned something new.